During the estrous cycle, the ovary is very active and its function can be divided into a follicular and a luteal phase. Primordial follicles consist of a primary oocyte in mitotic arrest surrounded by an uninterrupted single layer of rather flat follicular cells. Most of these follicles are located in the cortex of the ovary and are not developing, serving as a reservoir of oocytes in the ovary. At any given time, however, there are some primordial follicles which start to develop, but only a few of them ever grow enough to reach ovulatory size. During the follicular phase, some of the primordial follicles grow enough to become a primary follicle, which means that the oocyte starts to enlarge and what were flat follicular cells become a solid layer of rounded granulosa cells which multiply rapidly. At this stage, the zona pellucida also begins to develop. The zona pellucida is a layer of glycoproteins that surrounds the oocyte and prevent direct contact between the oocyte and the growing granulosa cell population. At the same time, the granulosa cells become encased in a basal lamina, which creates an impermeable barrier to blood-borne antibodies. By the time the zona pellucida is established, the granulosa cells have formed a multi-layer around the oocyte and an incipient antrum is formed among them. Outside the basal lamina, ceca cells, which divides in internal and external cecal cells, irrigated by a capillary bed, develops, completing what is known as a secondary follicle. After a follicle forms the antrum, it becomes FSH dependent. So, from now on, it needs FSH support for further growth. In the process of growing, the follicles start producing increasing amount of estrogen, which elevates the circulatory concentration of this hormone. As the secondary follicles continue growing to become Graafian follicles, they continue secreting more and more estrogen, which, at its peak concentration, trigger a massive discharge of GnRH with a consequent LH surge and FSH elevation. The LH surge triggers the process of ovulation, whereby the Graafian follicle ruptures, releasing the ova, and mediates a switch whereby granulosa and ceca cells become luteinized. That is, they cease to produce estrogen and start producing progesterones instead. This starts the luteal phase of the cycle. As a result of ovulation, what was the Graafian follicle becomes a mixture of blood, granulosa and ceca cells in what now is called the corpus hemorrhagicum. The cells of the corpus hemorrhagicum, now called luteal cells, commit to the sustained production of progesterone, and they proliferate rapidly using the blood material as nutritional substrate until the corpus luteum is fully formed. During the luteal phase, under high progesterone concentration, significant follicular development is inhibited. Thus, circulatory levels of estrogen remain low. If the ovary does not receive a signal that pregnancy has occurred, then the corpus luteum starts to regress, with a consequent decrease in circulatory concentration of progesterone. This reduction in progesterone concentration removes the inhibition of follicular development, allowing for new follicles to grow, therefore starting another follicular phase.